Blue-white screening by Kevin Godinez.
The process of blue-white screening is simple. In essence, it is composed of 5 steps. These steps are ligation, transformation, plasmid extraction, restriction digest, and electrophoresis.
The purpose of this step is to insert a target DNA sequence into a specialized plasmid called the cloning vector. Ligation reactions of target DNA samples can be prepared in various ways, including restriction enzyme digest and amplification by PCR. In this instance, we will shift the focus on the use of DNA sequences that have been amplified by PCR. The PCR target is amplified using GoTag DNA polymerase. GoTag produces an A overhang which behaves as a sticky end. It also permits ligation into certain vectors. Ligation of target DNA sequence into cloning vector utilizes a ligation mix. This ligation mix incorporates ATP, ligase, and buffer. The ligase enzyme creates phosphodiester bonds between adjoining nucleotides. The final result of ligation is the incorporation of the target DNA sequence into the cloning vector, thereby creating a recombinant vector. Transformation. E. coli are usually used as competent cells. One-shot cells are cells that have been made competent by chemical treatment. These cells are then incubated with divalent cation. After the treatment with the divalent cations, the cells are heat shocked. This produces a thermal imbalance on either side of the cell wall. This then forces the DNA into the E. coli cell. The transformed cells are then incubated in superoptimal broth with SOC medium to recover. Subsequently, this step is followed by incubation on selective media for blue-white screening. Blue-white screening. 2 genes are utilized on the PCR 2.1 vector for identification of recombinant cells, AMPR gene and LAC-Z gene. The AMPR gene conceals ampicillin resistance. The cells are grown on media which contain ampicillin, and only the cells possessing ampicillin resistance are able to grow on the media. The LAC-Z gene is used to determine the presence of a target DNA sequence in the PCR 2.1 vector. The LAC-Z gene possesses the B-galactidase enzyme. When paired with the X-gal, the enzyme cleaves the molecule into galactose and 5-bromo-4-chloro-3-hydroxyindole. This reaction spontaneously creates a dimer. The dimer is blue and thus makes the bacterial colonies appear blue as well. The colonies that appear blue were acquired from a bacterial cell that contained functional LAC-Z gene. Oppositely, the colonies appearing white were acquired from a bacterial cell with a disrupted LAC-Z gene. Consequently, white colonies indicate that the target sequence was inserted and thus transformation was successful. To summarize, when plated on media possessing X-gal and ampicillin, E. coli that has been transformed with a vector containing the insert will create white colonies, and E. coli that is transformed with a plasmid that does not contain an insert will appear blue. Before we can extract the plasmid DNA, we have to extract the genomic DNA. First, the cell must first be lysed open to release the DNA. Then we conduct the spin method, which precipitates the DNA using a lysis solution, which is usually cationic and attracted to the negatively charged DNA. And then ethanol is added to interfere with the bond between DNA and water, essentially making DNA nonpolar, and as a result, it precipitates out of the water solution. This solution is then captured using a silica filter in a spin column. Then ethanol buffers are used to wash the DNA to purify it. Then the DNA is eluted using purified water. Plasmid DNA extraction. Alkaline lysis. The alkaline lysis buffer is composed of sodium, SDS, and hydroxide. The SDS and hydroxide denature both the plasmid and genomic DNA. Afterwards, neutralization of the solution prevents damage to the single-stranded DNA. Right after the denaturing chemicals have been neutralized, the DNA begins to recreate the double helix. The plasmid DNA, being small, can easily reanneal, but the genomic DNA is too lengthy and reannealing becomes tricky, resulting in the DNA becoming tangled, thus keeping the complementary strands apart. Purification of DNA. The sample is centrifuged to isolate the plasmid DNA from the genomic DNA. The intertwined genomic DNA will complex with different intracellular proteins. This causes it to become heavier than the plasmid DNA. The plasmid DNA isolated in the supernatant goes through purification using the alcohol precipitation and spin column method. Restriction digest. We then check the DNA plasma to make sure the target DNA has been included. This can be done by digesting it with EcoR1, which cuts in the middle of the LAC-Z gene. Electrophoresis. We would expect to see our PCR target to be further down. Ideally, we should expect to see a linear piece of DNA and also a much smaller band below it. This would indicate that the PCR target was contained within the particular plasmid.